Unit 4 is cell communication and cell cycle. Cells communicate with each other in many different ways. One of them is direct signaling. Cells of multicellular organisms often maintain physical contact. Plasmodesmata and gap junctions allow chemical signals to pass through. The other way is through chemical signaling. This is when cells need to send signals over long or short distances. Short distance is often with cells of the same type. Meanwhile, long distance communication is often used to signal cells of another type. Endocrine signaling is long distance and travels through the bloodstream. The three stages of cell communication include reception, transduction, and response. In reception, a chemical signal called a ligand binds to a specific receptor protein in a target cell. When a ligand binds to a receptor protein, this causes a change in shape, which then causes transduction in the intracellular domain. In eukaryotes, a receptor protein would be a G protein coupled receptor. The signal comes and binds to the receptor protein, which then causes a change in shape. Change in shape allows for GDP to become GTP. GTP then gets activated and moves over to adenylate cyclase. After that's activated, it activates ATP, which then gets converted to cAMP, which then activates protein kinases. Cyclic AMP is an example of a second messenger. They relay and amplify the intracellular signal. Kinases are enzymes that transfer phosphate groups, which then relays a signal. Many signal transduction pathways involve phosphorylation cascades, where phosphate groups are passed along a bunch of times. A mutation that alters the receptor could lead to a different shape and function. This can lead to incompatibility, which can alter the signal transduction pathway. Signal transduction pathways can be activated or inhibited. Homeostasis is the maintenance of a stable internal environment. In order to maintain homeostasis, negative feedback loops regulate physiological processes. Meanwhile, positive feedback mechanisms amplify responses. Now, on to cell cycle. It consists of interphase and M phase. Interphase has three parts. G1 is where the cell is growing. S phase is where the DNA gets replicated. And G2 is when the cytoplasmic components are doubled. M phase has mitosis and cytokinesis. Mitosis ensures that there are two genetically identical daughter cells. It occurs in 5 steps. In prophase, the nuclear envelope begins to disappear and the DNA coils up into chromosomes. Then in prometaphase, the fibers begin to move the chromosomes to the middle of the cell. In metaphase, all of the chromosomes are lined up at the center. Spindle fibers attach. In anaphase, the fibers separate the double chromosomes into single chromosomes, which are chromatids. Then telophase happens where the nuclear envelope reappears. There are 2 nucleuses now. Then cytokinesis occurs, which separates the cell into 2 daughter cells. In G0, the cells are doing their specialized function. There are also checkpoints, which are pretty self-explanatory. Cyclins are proteins. They are involved in different stages of the cell cycle. Their concentrations can fluctuate depending on cell activity. The more cyclins, the more cell cycle progression. They are used to activate cyclin-dependent kinases, which are specific to the kinase that they activate. Cyclin-dependent kinases are involved in cell cycle regulation. They require cyclin binding. They phosphorylate substrates and promote certain cell cycle activities. When the cell cycle is disrupted, it can either result in apoptosis or cancer.